Welcome everybody to the 32nd edition of the Multiomics Reading Group. I'm your co-host for today, Cesar Miguel de Cordova. And today we have the absolute pleasure to have a Manu Saraswat presenting his work, which you now see on screen. Do you plan on giving any introductory words for our speaker?
Yes, as usual. Okay, thanks Cesar for the introduction and also the recording. Okay, let me give some very short introduction about speaker today. So Manu, he completed his PhD in computational biology at EMBL and German Cancer Research Center in Heidelberg, Germany, with Oliver Steger, where he developed interpretable deep learning frameworks connecting genetic variations to cellular states and disease. His work spans inferring gene regulatory networks from single-cell multi-omics to building sequence-to-expression models for personalized genomes and linking genetic variations to cellular phenotypes at population scale. During his PhD, he also spent time at Genentech exploring transformer-based approaches for regulatory genomics. He's now a postdoc fellow with Dana Pear at Memorial Sloan Cancer Center. Well, Dana is super cool, integrating spatial, temporal, and perturbation data to model tumor immune interactions, with the broader goal of building AI systems for biology that can both be powerful and interpretable. So let's welcome Manu. Thanks for accepting our invitation, and yeah, it's all yours.
Thank you so much for a very lovely introduction, and I'm very happy to be here. And I'll talk about one aspect of my PhD work, which focused on inferring gene regulatory networks from single cell multiome data and how we can use those networks to understand plasticity in diseases. And in particular, I will talk about glioblastoma, which is a highly plastic disease. So we're talking about plasticity, and then I'll start off by saying that it's a very fundamental process for biology. And when you look at a stem cell differentiating to say neurons, this plasticity is progressively going down. A more initial progenitor state is becoming more mature, and that happens through actions of transcription factors which can bind to cis regulatory elements. And these cells are basically kind of locked into the state. The plasticity is kept under control. But what happens in cancer and other diseases is that this plasticity can go unchecked, it becomes unlocked, and it's now recognized as one of the hallmarks of cancer. And thereby, the cancer cells have the ability now to evade, say, treatment or immune responses or therapy, and be able to have metastatic potential, like a lung cancer cell can invade other organs, and all of that happens by this unlocked plasticity. So it's very important to understand just from a normal developmental point of view, but also to understand diseases in more detail, like how is this plasticity occurring, what are the underlying molecular players like enhancers, transcription factors, which govern this plasticity control. And luckily for us, in the last years, with the advent of single cell omics and single cell multi-omics in particular, we now have a better hand at answering those questions of what is the underlying regulatory process of plasticity. So on one hand, what single cell RNA-seq gives you is like these different states which potentially are very which were not captured before in bulk data sets, and might be rare and intermediate, and then you can do things like pseudotime and so on, and understand like how does one state go and differentiate into the other, and you get like these snapshots of different states. But also alongside with multi-ohm data, you might also have attack-seq information available, which is telling you not just which genes are expressed, but also which regions of the chromatin are accessible. And with that, you can get information about like, okay, which could be the underlying motifs which are accessible in these particular states, thereby which TFs could bind, and then you can get a better hand of gene regulatory networks, or basically transcription factor to gene networks, which are coordinating these different biological processes. And to go from this multi-ohm data to the GRNs, as we like to call them, there have been a lot of computational tools which have been developed in the past years, and you would have heard some of them: Scenic Plus, Cell Oracle, Pando, Granny, there's Dicties, and others as well, And they work quite well actually with the type of scale of the data that was available at the time when those tools came out. But now with like increasing data set sizes and the type of questions we want to answer, these tools have a lot of limitations. And before I go into those limitations, I want to like just quickly give you an overview of how these different tools work and what sort of signatures they try to capture from multi-ohm data. So because you have ATAC-seq information available, you can do things like some sort of co-accessibility pattern analysis, which is telling you that, okay, in a given cell state, these sort of enhancers or regulatory elements are accessible together and they're working in conjunction in modules. You also have information, as I already hinted, about which transcription factors could bind to those regulatory elements. And because you have both accessibility and gene expression from the same cell, you can start to do some correlation sort of analysis to link the accessibility of a region with the expression of genes which are surrounding that region, and then get these enhancer to gene links. And of course, you can also look at the co-expression of TF and gene, and with the idea that like if a TF is expressed, gene is expressed. If a TF is not expressed, gene is not expressed, then probably this TF is regulating that gene, and vice versa, that it could also act as a repressor, that it might have a negative correlation. So all these different tools kind of like distill these different signatures in one way or the other. The exact way in which they do it might be different, but like this is essentially all these models try to do, and this is also what I will do in the approach I will show in a bit. So coming back to the models and their limitations, so one thing we are interested in is like what is happening in these intermediate states. So if you want to understand this transition, and if you want to understand what sort of GRNs are active in it, the current approach when you take these models would be to just take cells from these different like clusters and then make a network for it. Now, this is obviously ignoring the beauty of the continuity of the single cell data, because you have to define these clusters arbitrarily, and sometimes you don't even know where the cluster end or starts. But also, you're not sharing information, that you might have a big data set, but you are only looking at a handful of cells when you make a network like this. Of course, you can also make the network for the entire data set using all the cells that are available, but then obviously you get one giant global network and you don't really understand what are the differences in those networks across these different states. So you're discretizing the information, which is not ideal for single cell. But also, if you are interested in some very rare cell state, again, like if you say I just have 50 cells, none of these models have the statistical power to get something meaningful out of it. Like the correlations will not make a lot of sense. You could argue that, okay, I could just sample a lot more cells, like I have data set of millions of cells, and maybe this cell state becomes larger. But the current tools actually are not scalable to the scale of like millions of cells, even a few hundred thousands of cells, to be honest. So with these limitations in mind, we wanted to develop a tool for inferring gene regulatory networks, which is very scalable, works with data sets of millions of cells, but also more importantly gives us continuous gene regulatory networks in a biological system. So to achieve that, during my PhD, I developed this tool called SCDORI, or Single Cell Deep Multi-Omic Regulatory Inference. Now, from a broad overview, this tool is an autoencoder, which means that for a single cell, it takes its accessibility profile and gene expression profile as input. It reduces it to a dual-dimensional representation of topics, and then reconstructs the original profile of a ATAC-seq in gene expression and RNA-seq. So we know autoencoders have been used a lot for single cell data, for RNA, for ATAC. We have peak VI, SCVI, multi-VI. So by using autoencoders, we can train these models on mini batch. So you're solving the problem of scalability now, because now you can train these models on arbitrarily large number of cells because you don't input everything into memory at once. You can do mini batching. So you've solved the problem of scale by having this formulation in autoencoder. But we still don't get the information about the GRNs. And to achieve that, what I did in this approach is, instead of having a non-linear decoder, which is usually the case, that you have a neural network which is fully connected, which takes the lower dimensional representation and reconstructs the original profile, we have a set of interpretable linear decoders. So we have different linear matrices which try to reconstruct the data in different ways, and in doing so, they learn the GRN. And I will talk about it, what I mean by that. But these different decoders essentially capture the different signatures of GRN I talked about, whether it's co-accessibility, TF binding, enhancer gene link, CF gene correlation. All of that happens in the decoder. So in the next slides, I will talk about it. But what you can take away from this slide and this approach is that what ultimately SCDORI gives you is that each cell now is a linear combination of topics. So there's a softmax over here, and each of these topics themselves is a gene regulatory network. So each of these topics is its own network of transcription factor to gene links. If the TF is not active, all the downstream links will be 0. If it's somewhat active, it will have some links which are positive or negative. And then for each cell, you can now interpret that, okay, this particular GRN is active to like this degree, like it's 0.6. And this one is also active, but to a lesser degree. And some other GRNs are more or less active. So you get like this continuum of GRNs basically for each cell. And then you go away from this problem that like you have to discretize the data or work at a cell type level or and so on. Okay,
So
are there any questions?
Yes, I have a really quick question regarding how you choose the number of topics. This
Yeah.
is sort of implicitly tied to the types of cells you have in the model.
Yeah, I think it's more tied to the number of cell types and cell states you will expect in your data set. So that is a good starting point to begin with when you choose the number of topics. But because we put a softmax layer for each cell over these topics, there is an implicit regularization that happens. So even if you start with a larger number of topics, what we see, the model basically deactivates certain topics. It just doesn't use them. They're all 0 everywhere. So you can start with a slightly larger number, and then the model will choose whatever is necessary, and remaining will be 0. Of course, if we choose a lot of topics, you will increase your computations, because I will show you in the next slide, like the computations happen per topic. So there will be topics which are computing things, but they're not being actually used because there are zeros everywhere. So it's just an additional overhead. But yeah, a good starting point will be certain number of cell types per plus some extra topics.
Thank you. That's a great precision. Just to have the number in your heads, for this paper, you guys go for like 30 something. Is that
Yeah,
it?
exactly. So yeah, that's correct. So the full picture is that we trained with 50 topics, and then around 30 were active. So
Okay.
then 20 were basically like not used exactly. Yeah.
Wonderful. Thank you.
Yeah. All right. So this is the main takeaway. If you have a multi-ohm data, single cell multi-ohm attack RNA data, what SCDORI will give you is these gene regulatory components or GRNs, and each cell as a combination of those. I will show examples on cancer data set, but this model is really applicable on any multi-ohm data you would want to understand and get these networks for in a continuum. All right. So diving into the details of how this actually happens, it's a complicated model, but I want to give some snippets of how that is happening to give you some flavor of what I mean by linear decoders and how that happens. So let's look at the accessibility prediction. So so we have like bunch of different decoders, like bunch of different matrices. So there is one matrix which is topic stamp speaks. So if you take the output from the encoder, which is the cell stamp topics latent space, and you multiply it by the topic stamp speak matrix, you basically just get the reconstruction of the peaks. It's very simple. But looking at this topic to peak matrix, this is effectively this co-accessibility pattern matrix. It's telling you that in a given topic or a cell state or a cell type, for simplicity, these are the set of peaks which are accessible. And then if you're a given cell, like this topic is active, then this peak should also be accessible. So that's like the simple idea. We also reuse the same matrix and learn an additional matrix of peaks times genes. And then if you take the product like this, you have cell stamp topic times peaks times genes, you get the reconstruction of cells times genes. What this is just telling you is that, okay, in a given topic, these peaks are accessible. And the model learns that what sort of genes are those peaks regulating. So you're trying to learn this relation of peak accessibility and gene expression in this way. Of course, there's some constraints on this matrix that the peaks and genes have to be on the same chromosome. On a different chromosome, they are set to 0 by default. And there's also like a distance-based threshold that the peaks have to be within a certain window. Those are all hyperparameters one could choose. So it's like a biologically guided, mechanistic way of achieving, like of instilling like this particular aspect of gene regulation into the reconstruction task. These are only the two modules. The actual model looks like this. So it's going to take a lot of time to explain what's going on here, but like I will just also talk about like the actual gene expression very briefly. And if we have time later, I can talk more into detail like how that is done. So far we have gotten the peak to gene matrix, and then also topic to peak matrix. So what then we do is we try to learn a peak to gene matrix, which is topic specific. What I mean by that is that this matrix is just telling you that for a given gene, these sort of peaks, these like 4 peaks or 3 peaks, whatever, could be regulating this gene. Now, in different topics, like out of those 3, like some of them might be accessible, some of them might be not. So, but if you're looking at one particular topic, you're only interested in the peaks which are accessible there. So very simply, we just do an element-wise product of this topic stamps peaks matrix, which we have already learned, with this peak to gene matrix, like kind of like a mask. So for each topic, you get a new version of this peak to gene matrix, which is telling you you're just hiding out the peaks which are not accessible in that topic, very simply saying. That's all that is happening here. You can take these matrices, multiply them by a peak to TF matrix, which is telling you which TFs have a motif and could bind there. Like we actually use something called in silico chip-seq, which is a bit more involved, which also uses TF expression and peak accessibility apart from the motif. But when you do the product of these two, you get a TF to gene matrix per topic. This is just telling you that, okay, in a given topic, these peaks are accessible. We know that these peaks are linked to these genes, and we know that these TFs are binding to those peaks. Thereby, this TF could potentially regulate this gene. This is what is happening here. Then on top of it, we also use information about TF and gene correlation of expression. So the idea is to learn a TF to gene matrix per topic and then use the TF expression to predict gene expression. You learn a matrix of these TFs are regulating this gene. You use the TF expression to predict the gene expression, and that's your backpropagation loss. And iteratively, you will refine your network of TF to gene so that you get better and better reconstruction of the gene expression profile. How the model actually works is that you can do everything all together end to end, but to reduce the amount of computation and make the model train a bit faster, I first train like this top part, and then in the second step I freeze everything, and then I just learn the TF2 gene links and predict the gene expression. This is how operationally the model works, but you have the choice in the code that there's a flag that you can train it all end to end. Now, this was the activation part. The same thing happens for depression as well. Everything I said, but reverse. So a TF binds, it closes the chromatin, gene expression goes down, and then this is how we model repression, and all of the like both the things happen simultaneously. So you get a net effect of whether TF has a net repressive or net activator effect in different topics, and that's how the model actually works. So I said I'm going to talk about the details later, but I already mentioned quite a bit of it now. I would want to go through like rest of the talk as well, but since I've talked about the details now, maybe I will stop here and ask for questions if you have any about the modeling.
Right. So would it be fair to say that you're jointly learning activation and repression?
Yes, that's correct.
So given that, how could you ensure that the model is actually learning true regulatory biology and not just correlating between these active and repressed modes, let's say? And was there any sort of validation you carried out?
Yeah, I mean, so at the end of the day, a lot of what we do in these models is correlation, but by enforcing certain biological constraints, like you try to like make sure that like you try to capture as much causal information as you can. In this case, that would mean that the peaks and genes cannot be arbitrarily like on different chromosomes. They should be constrained within a certain window. The TF and gene, if they are correlated, the TF also needs to bind to those enhancers. It needs to have a motif, so that also ensures like another layer of information. So all of these constraints kind of help. And the model basically picks up like what is the larger effect. Like you model both simultaneously, but you see basically for a given TF gene pair, which has like a larger effect, and then that's the net effect the model learns. But of course, like you cannot fully say that like this is, you're working with observational data, you're trying to infer TF to gene links, so there's only so much you can do. And of course, to really know whether a TF to gene link or the peak to gene links we have inferred from the model are true, you need to go and do some experiments. And that is what we do to validate our approach. So in the paper, you can also read those details, but just to give you some idea, we look at, for example, say Hi-C data, which is chromatin conformation, and CRISPR-I of enhancers in different in cell lines. and so on. And we apply std, infer these links, and then we see whether we are able to recapitulate what it's there from the biological validations. And we also compare it to other methods, in particular Scenic Plus. And you can do the same for TF to gene links as well. You can take CRISPR knockout data of a TF and then see which genes are going up and down, and see how well the genes which this model predicts are going up and down, are they like aligned or not. And we do that for certain transcription factors, and we are doing it for a lot more as part of the revision process. Yeah, so hopefully that gives you some idea of the validation process of this.
Yeah, that's fair. Wonderful answer. I'll be looking at that closely for sure. Thank you.
Yeah. Cool. Any more questions?
Yeah, I have one.
Yeah.
Regarding the module 1 and module 2. So in module
Yeah.
1, you learn the topic by peak, right?
Yeah.
And then you decompose this matrix topic by peak into so you don't decompose. So you learn also matrix C to have
Yes.
topic by topics peak time peaks gene gives topic gene. But this is an arbitrary order, right? You could have done the opposite. First, learning the gene in module 1, and then decompose into and then learn the peaks by the gene by peaks, right?
Yeah, so it's happening simultaneously. So it's not that I do this and then do this. It's all happening together. So like these matrices are the same and they are learned together.
Okay.
So you have both
but
the optimizations happening at the same time. So you predict expression, you predict accessibility, and the reconstruction is a combination of both.
Okay, but if in module 1 you have learned instead topic by gene, and in module 2 you had topic by gene and gene by topics, would it have changed the result?
So the idea is that we're not learning a topic by gene. Like, so this is an assumption, maybe like that's what you're getting at. So this is an assumption we have made, that we are trying to use accessibility as a predictor for gene expression. That's what we are trying to do, because that's like biologically, that's what we think, that okay, there is an enhancer which is accessible, and when it's accessible, certain TFs could bind there, and then they can regulate the gene expression. So this is like the causal chain of events we are trying to model, and that's kind of what's reflected here, that you learn what is accessible in a given topic, and then you learn that based on their accessibility, which genes could they potentially regulate. And this is like what we are modeling here, like what we call module 1, module 2, that is arbitrary. Like, so this is arbitrary because this is all trained all together just for like explanation. Both these things are trained end to end. Yeah.
Okay, no, but that makes sense. That makes totally sense. Okay, and I'm confused a bit about stage 2, because you end with the topic by gene matrix, right? But at stage 1, the topic by gene matrix is actually the topic per peak times the peak per gene. So at stage 1, it's fixed. You already have the topic by gene matrix, right?
Yeah.
But so it means that in stage 2, you end with a new topic by gene matrix.
Yes.
But so therefore, it means that if you want to get the same topic by gene matrix at stage 1, you would need different matrix B and matrix C.
Yeah. So maybe I would say that, like, so the end goal is to predict gene expression, reconstruct gene expression. I think the way we are constructing this gene expression is in like 2 different ways. Like in here, the idea is to use the accessibility to reconstruct gene expression. And in this stage 2, the idea is to use the TF to gene matrix to reconstruct the gene expression.
Okay.
But using the components of what we have learned on top. So these things are learned and then they are kind of fixed. And then on top of it, you learn the TF to gene matrices, which are derived from these matrices. And you again do the reconstruction of gene expression. And then that's how you train this, if that makes sense.
Okay, it's more clear now. Okay, thank you.
Yeah. Cool. Any more questions? All right. If not, then just this message that you can use it to decompose your single cell multi-ohm data into continuous modules of gene regulatory networks. And as I said, you can apply it on any single cell multi-ohm data you would be interested in. And check out the code. And then if you have questions, you can also reach out to me later. All right.
Just
so coming,
just a little context before we go further. Like, have you seen any systems, any sort of developmental data sets that are interesting that you have applied SEDORI to that where it just works, let's say?
Yeah, definitely. I mean, so when I was developing this tool, I was not working on cancer. So the data set I was working on was a mouse gastrulation data set.
Alright.
So the first thing I applied to was a mouse developmental data, and it worked quite well there. Like, because we also, well, it worked well, and by well I would say that we were identifying topics which were capturing different cell states, and the transcription factors which you were capturing made a lot of sense because developmental is very well studied, so you know different tiers for different stages. So yeah, it definitely works there. We are also applying it on other cancers now. We are applying it to data sets of heart and brain organoids, which is not like normal development, but it's also development in some other sense. And also applying it to like disease versus healthy control patient data multi-ohm from like ALS disease from brain samples. So it's like different contexts we're applying and then seeing like very encouraging results.
Very nice. And for example, just brainstorming a bit here, if you switch the encoder for something a bit more specific and then got the decoder only component of SEDORI, do you think that would work? Maybe, maybe not, maybe a fun experiment to try. What kind of modifications would you sort of encourage people who are into model building do or look into?
Yeah, I mean, are you asking like what's other like new sort of developments one could do over top of SEDORI, you mean? Or
yeah, or like for example, which kind of encoding decoding schemes would be used for multi-omic data more broadly, let's say? Like if you had to suggest extensions to SEDORI or maybe alternative ways of creating these rich latent spaces that you could then perform.
Yeah, totally. I mean, so there is one aspect which I'm still thinking about what is the right way of modeling it, is that if you have like data from like say thousands of people, so like if you want to apply it on a population scale data, and the questions you're interested more broadly is like how does like you are interested in like genetics and like population level influences on gene programs and neural networks, like how would one adapt like a model like this for those aspects. Another modification would be that if you have like data from say perturbations on top of just wild type data, like how do you adapt like this encoding scheme to reflect also, like the idea would be to have parts of the model which are kind of like basal or like static, like this is like underlying biology, and then you learn perturbation specific effects which are just changes to those networks which the model has learned. So like one could think of like interesting ways of encoding that aspect into the model if you're just interested, okay, what does this knockout do to my networks across different cell states. So those are some of the questions I think I would find interesting. What I am doing, and I don't know if that's what you are like, if that's like the domain you were referring to or interested in, but like what I'm also doing now in my postdoc is to take some of these ideas and also extend them to the spatial context. So now we are getting data sets in which you have multi-omic single cell attack in RNA, but from the same like not from the exact same cells, but the same tissues. You might also have spatial data. And then how do you leverage this information of cell-cell communication and the downstream gene regulatory networks, which we are capturing here? Like, can we make like a causal cascade, like extending it to the spatial level, that you have the signaling molecules which basically activate certain transcription factor networks and so on. So that's another extension I'm working on now.
Very nice, thank you.
Cool. So let's jump into cancer now, and this picture is, as I showed before, is reflecting that cancer cells regain this plastic nature, which allows them to do all sorts of things which are undesirable from a disease and treatment point of view. And one particular plastic cancer is glioblastoma. Highly aggressive plastic cancer, no treatment possible. Median survival time is just from diagnosis just like 15 months. So basically there's no treatment. And one reason for that is this its highly heterogeneous and plastic nature. And what existing studies of single cell RNA have already showed is that the glioblastoma cells like exist on a continuum of 4 major states, and these states are they have programs which are capturing like the healthy brain cells which you would find in development or in adult cells. So you have glioblastoma cells which are AC like or astrocyte like, you have cells which are gliosis hypoxia like, you have cells which are neuronal like, and you have cells which are oligodendrocyte progenitor like or OPC and neural progenitor cell like or NPC like. But really, every cell exists somewhere on the continuum. Like you have some cells which are more reflecting one identity or the other, but all cells share some identity with other programs as well. And on top of it, the cells have the ability to transition between these states. So an AC-like cell can become OPC-like, and OPC-like can go to neuronal-like, and so on. And what we wanted to answer is that what is the intrinsic regulation mechanism which these cells use to perform all these different transitions? Because if we have a hold on those mechanisms, then we can potentially like try to control that mechanism so that the cells do not go as wild as they're doing in a normal scenario. And to tackle that problem, we collaborated with Omar Barakta's group at the Wellcome Sanger Institute in the UK, who generated this huge atlas of glioblastoma consisting of 1 million nuclei, for which we have single cell multiome attack and RNA from the same cell. It's coming from 12 different patients, multiple sites per patient tumor. So the largest multiome data set which exists for glioblastoma so far. And we annotated these different cells and recapitulated what I showed you before. This over here is the cancer cluster. You have the neuronal-like cells over here, the OPC, NPC-like cells here, AC-like cells here, and gliosis hypoxia-like cells here. And then these are the tumor microenvironment healthy cells, which are neurons, OPC astrocytes, and some immune cells as well. This is the landscape based on just RNA that we know the markers from previous studies and we can do the annotation. But what this atlas additionally allows you to do is not just look at the expression of like which genes are expressed in different states, but also which genes are potentially accessible, like how does the chromatin look like? And chromatin actually gives you a window into this plasticity already. So the idea is that if a gene is not expressed, but it's already accessible in a given cell state, then that cell potentially has it has a potential to transition. So these green cells have accessibility around the orange genes, which are not yet accessible, which means that in the future, maybe this gene could become expressed, and then these cells can start to transition into the orange cells. And we use this like concordance or disconcordance between the attack and RNA in a given cell as a measure of epigenetic plasticity. Very simply, we just say that, okay, if the expression and accessibility are aligned, then the plasticity is low. And if there is a lot of confusion that you have accessibility around genes which are not expressed, then there is more plasticity, more epigenetic plasticity. And this idea was taken from this paper from Dana Pierce's group. This is just done using like a linear model, very simple logistic regression. So when we do this approach, what we very clearly and strikingly see is that the TME cells, the healthy neurons and OPC astrocytes and immune cells, they basically have very low plasticity score. And this cancer cluster is just like lighting up like anything. You have very high plasticity in these cells, very clear distinction. But even within these cancer cells, there's a striking difference between different states. So this neuronal-like state has a significantly an abnormally low plasticity compared to other glioblastoma cell states. So there's something special happening in this particular neuronal-like state, and we wanted to understand like what is happening here and what is happening here. Like why are these cells so plastic? What are the regulators? And to understand that, we take help of Acidori and apply Acidori to this entire multi-ohm data, which gives us these multiple networks. As I already mentioned before, we started with 50 topics, and 31 topics, if I remember correctly, were active to a certain degree in at least some cells. So, and when we visualize those topics and see what they're doing, we very nicely see that you have these different topics which are capturing the broad cell states I talked about. Topic 24 in neuronal-like cells, 34 in OPC, NPC-like, and then you have topic 20 AC-like and 19 gliosis hypoxia. But coming back to this point of like continuity and transitions, we also see these topics which are intermediate between states. So 49 is capturing these transitions from like neuronal like states. This is between neuronal like and OPC like and so on. Like, so there are like topics and gene algorithm networks which are capturing intermediate states, which might be harder to pick up if we were just to discretize the data into clusters. And with a Sidori, you get for each of these topics its own set of genes and peaks and transcription factors. Some of these are known from literature. You can take these TFs and start doing validation in the lab and then see whether overexpressing these TFs makes your cancer cells go into one state or the other. And those are the sort of experiments which are being done in the lab by us as well. Maybe I will skip these two parts and come to this real question of how does one particular cell state transition into the other. So after applying Sidori, we get like these different networks. So you're getting like two information here. So if you want to go from a source topic to target topic, you have the information about which sort of transcription factors are active here and what are their downstream influences. And in the target topic, you also know which TFs are important. So I will give you an analogy of like reprogramming. So say if you want to reprogram fibroblasts to neurons, typically what you would do is you will overexpress certain transcription factors, which are neuron specific. You overexpress the master regulators of the state you want to go into. And we use a similar idea that if a state wants to go into the other state, it has to kind of activate the master regulators of this state. So with these networks, you can identify which are the topic regulators or the master regulators of each topic. Basically, the topics which have like a maybe here like the strongest downstream influence, like set of TFs which target a lot of downstream genes are the master regulators or TRs or topic regulators. And the idea is that if a source topic has to go into a target topic, it has to activate these topic regulators. And how does it regulate those? So we have the information about the GRN. So we can start looking at the TFs here and see whether their downstream targets are enriched over here, whether these TFs can bind to the chromatin, to the enhancers and genes of these particular transcription factors. And we get all this information from Sidori. Because we have multi-ohm data, we also have information about epigenetic priming. So you can also look at whether these TRs are already accessible here. I already talked about this epigenetic plasticity. They may not be expressed here, but if they're already accessible, then there's a high potential for these to become activated. And we combine these different like signatures of whether these TRs are epigenetically primed here, whether there are TFs which could bind to the chromatin and enhancers of these region and activate them. And then also like looking at whether this particular TR, like how influential it is into one score of topic activation potential. It's like a mixed complicated concept, but very simply, you just want to go from A to B, and to go from A to B, you want to activate B, and you try to identify or quantify basically how easy it is to activate B based on these general networks. And when we do this, what we see is that for certain topics, this potential to transition is very low. And for this is particularly true for the neuronal like topics, like you barely have some links to other states. And on the other hand, you have like these really like this grid of OPC and PC like topics, which can transition to all other virtually all other different states and topics. And you can combine all of this information at the state level, because in each state you can have multiple topics. And when you look at this map at the state level, you get this highway of potential transition where you have these OPC and PC like and AC like states, which have a lot of very strong links to other states, like they have a high potential to transition, whereas this neuronal like state is basically having very, very low weak links. I talked about activation, but you can do exactly the same thing for repression as well. Whether there are TFs which could bind to other TFs in state B and then close the chromatin and basically reduce the expression. And if you do that, you get an opposite picture, that there is a widespread, very strong repression happening in neuronal-like state. So these cells, basically, their chromatin is closed and they are actively blocking transitions to all other glioblastoma states by closing their chromatin and making sure that those other transcription factors of these states are not becoming activated. Combining these two into one map, we get this net transition potential, which is just giving this picture that you have these high plasticity states like OPC, NPC-like, and AC-like, which is also what we saw before, which can transition easily to others. And whereas at the bottom of this plasticity hierarchy is the neuronal-like state, which we also saw has low plasticity, and it's doing so using repression. It's actively blocking transitions to other states. That's what we model. Is there a question? Okay.
Yes, a really quick one from my end. This is just another like sort of perspective, let's say, question. This is pretty telling, right? You're most definitely capturing plastic states. But if you had to add one data modality, just one, any of the many that exist, to further augment the resolution of the plasticity happening in GB, what would it be?
Yeah. I would say like some other histone marks would be useful, like some activator or repressor marks. So accessibility is giving you some picture into the regulation, but not all accessible regions which are linked to a gene are enhancers. So I think on top of it, if we add this another layer, that okay, these are truly enhancers or activating and repressing like regulatory regions, I think that would like add additional value to these analysis, definitely.
That's great.
Yeah.
Awesome.
So these are still all predictions. So what we also wanted to like do is some sort of validation. So in the huge atlas, apart from multi-ohm data, we also collected spatial data, VCM. And our hypothesis was that if cells can transition easily between each other, if two states can transition easily between each other, you will also see those cells in more spatial proximity. Whereas if a state is less likely to transition to other, like you will have more distances between those two states. So basically we quantified like distances between all these pairs of states. And what we see is that indeed the neuronal like state is the most constrained. It has the maximum distance to all other states, and all these other states which have more easy transitions, they are like more like spatially they co-occur. So using the multi-ohm data, which is totally disjoint from the spatial data, we are already kind of explaining like the tumor architecture, the spatial tumor architecture based on GRNs, which I think is pretty cool in itself, but also is giving us some insights and some validation into some of these predictions we have done. So this is all very nice and cool. And I want to summarize this part of the talk that so far we have looked at the epigenetic plasticity of glioblastoma cells, that certain states have more accessibility around alternate states, and others like neuron like do not have that capability. By applying Sidori, we can get some insights into what sort of gene regulatory networks are active in these different states, but also use those networks to kind of get a transition map, which telling like a highway or a barrier to these transitions, like these are easy, these are difficult, and giving you some insights into a plasticity hierarchy. And the last part of the talk is, can we use these insights about repression and plasticity hierarchy to kind of control the tumor growth? And the idea would be to use these repression mechanisms which we have identified before, that neuronal-like states are actively blocking transitions. So with Sidori, we can also identify what are the repressor TFs. And one TF which we identified from Sidori is MIT1L. So this TF is a known repressor in neurons. So actually its role in neurons is to make sure that neurons stay like neurons. It goes and it binds to the master regulators of all these other different cell types. It makes sure that the genes from muscle are repressed and shut down in neurons. So neurons do not become something else. So it has this like safeguarding role in healthy neurons. And in GBM, what we predict is that this transcription factor is being repurposed, and instead of blocking other alternative states, it's blocking other alternative GBM states. So it's binding to the transcription factors of AC-like, OPC-like, and gliosis-like states, and it's blocking their expression. This is the MIT1L expression, like very highly specifically expressed in neuronal-like cells. And this is the expression of the genes which you predict MIT1L is repressing. And you see that it's basically blocking the repression of other GB states. So the idea would be that we take GBM cells, and if we overexpress MIT1L, then you would constrain the transition of these cells to only neuronal-like, because all these transitions will be blocked. So this initial plasticity and flexibility which GBM cells have to really go anywhere, this will be constrained, and then you will make sure they are only restricted in one state. And that potentially is one way you could tackle glioblastoma from a therapeutic point of view as well. So we went ahead and did this exact experiment, took patient-derived GBM cells, basically overexpressed MIT1L, did attack-seek and cut and run on top of it to basically identify which regions of the chromatin are becoming open and closed, and where does this transcription factor bind. So one thing very strikingly we see is that when you overexpress this transcription factor, majority of the peaks become closed, which is in line with this repressive action. And the peaks which are closed are basically from these AC lagging dioses like hypoxia topics, like what you predict from the model that's really happening, that it's closing the chromatin of alternate GB states. But also it's not just a correlative indirect effect. It's really happening because we have the binding of this transcription factor in the master regulators of all these other states, I'm showing here. So these things are picked up by a Sidori that they are the master regulators of OPC-like, AC-like, and gliosis-like, and we see that MIT1L actually binds to the chromatin of these genes and closes them. There's also some activation happening in neuronal-like state, which makes sense, but broadly, it's a repressive action. So from a survival point of view, we transplanted these cells into mice as well, and compared that to cells which did not have this overexpression. And when we do these experiments, we also see that the total cell count, like the growth of the tumor, has reduced in MIT1 overexpression, but also in general, the mice live longer with this overexpression in the mice which have the overexpression of MIT1 happening. So really, we are using the insights from multi-ohm data about plasticity and its control, and kind of going towards a more therapeutic translational angle, and using those insights to hopefully in the future have better therapies. Now, in the next phase of this project, we are kind of seeing how does this overexpression perform in conjunction with existing therapies, like typically you would give radiotherapy and chemotherapy. So now the idea is that you do this overexpression, constrain cells to be neuronal-like, and then do give them therapy, as opposed to giving therapy to all the cells. And maybe that gives a better survival advantage. That's yet to be seen, but that's the hope. And I'm at the end of my talk, and I would like to thank all the great collaborators who made this project possible. And specifically, I would like to thank Laura from Moritz Mal's group and Elisa from Moritz Mal's group. Laura helped in a lot of the validation, and also the like a lot of the competition analysis we did with GBM. I wanted to thank the Omar Berekta's group, Grant, who did the pre-processing of the data, Tanya Fani, Oliver, who generated the data, and then members of the State Lab, which were very helpful in developing Sidori and all the discussions with the machine learning aspect of this approach. And thank you all for listening. This is the final summary of this project, and this is a preprint. I don't have. I should also put the GitHub link for the project, but you should be able to find it on the preprint as well.
Thank you, Manu, for the absolutely awesome presentation. If you do have that GitHub link, we can put it in the YouTube channel, like when the talk goes up, and that way we'll have a way to post it for everybody to see. And with that, after thanking you for that wonderful talk, we would like to open up the floor for additional questions from the audience. I know I've been asking questions all throughout the talk, so if anybody else would like to ask something to Manu, this is the moment to do it.
Yeah, I can start, even though I also ask questions. I have so many questions. It was an amazing talk. Thank you a lot. But just choosing two of them, one very operational. How do you preprocess your single-cell RNA-seq and single-cell ATAC-seq? Because you have lots of peaks if you work with ATAC-seq data. So
Yeah.
can you give more details what you did?
Yeah. So yeah, choosing attack peaks is always a tricky point. So in this particular case, and like a general strategy, what I use is that I first select the genes which are highly variable.
Mhm.
That could be whatever number your GPU allows. Like a lot of the choices we made in this particular paper were based on the GPUs we had at hand. I would have liked to work with larger set of peaks and genes, but that's what the GPU allowed. But you start with a set of genes, 4,000, 5,000. If you have certain genes and TF which you know are relevant for your data set, you keep them. And then what I do is I take the window around those genes. That's a user specified window. In this case, it was 150 kilobase pair around the gene body. So you take only peaks which are within those windows, because that's what you're modeling. You're modeling accessibility to expression link, and then that already reduces quite a bit. On top of it, you can further do like highly variable peak and so on sort of things to further reduce the number of peaks. If you have some, say, bulk data or some other auxiliary information about these sort of regions are relevant in my biological question data set, you can just make sure that you include those regions by default.
Mm.
But yeah,
And,
that's the
for
short
you,
answer. And there's no,
yeah,
sorry, just one point,
yeah,
like there's no
sure.
pre-processing on the exact counts. So the counts are raw counts.
Okay. And you keep a window of 500 nucleotides for the peak, or do you have variable length peaks?
I think I just use Archer, which
Okay.
constructs like the peak gives 500 base pair
Yeah.
peaks. So
Okay.
I haven't like looked into the influence of it, but like having a uniform set is fine.
Yeah.
And it could be a larger. You could have 1,000 length peak as well. I don't think it would really
Okay.
matter so much. Yeah.
Okay. Great. My second question is: do you plan to work on temporal data? So to see how the topics and the GRNs evolve in time.
Yeah. That's definitely a plan. So far, what we have done is like you could take a temporal data and apply the model, and then you can see if you don't put any constraints what happens. So even then what you see is that there are topics which are activated at different time points. That's like the most straightforward naive way of just using it, but it's not really like capturing the actual temporal information. So I think in reality, like if you want to really model it systematically and more principally, then you would put some sort of smooth transitions in those topics that if two cells like two time points if close by, then their topics should be like smoothly transitioning. I don't know, like putting
Yeah.
Gaussian process or some sort of constraints like on that. I think that would be
Yeah.
like the way to go. But
Sure.
I'm not
Yeah.
actively working on that aspect, but that I think is a very interesting extension
Yeah.
of the model.
And even just to confirm if the unconstrained modeling gives valid conclusion. If you
Yeah,
model the transition between the GRN,
yeah,
does it give the true transition that you observe? But
yeah.
yeah, amazing work. Thank you a lot.
Thank you so much.
We have a question from Chen. Please go ahead.
Hi, hello. Can you hear me?
Hi. Yeah.
So yeah, so I have a question on the model. So you use
Mhm.
this autoencoder. So it seems like you found the GRN, like those genes you found from autoencoder are like this some genes that have significant expression. Am I understand correct?
In the GRN, you mean, or?
Yeah. So you're outputting those GRN from this autoencoder. Can you elaborate on that? Yeah.
Yeah. Basically, so the idea of the model is that you learn the GRN,
Mhm.
and which is the topics to gene matrix, and then you use the TF expression to predict gene expression using this, like in a linear model, basically. So you learn that for this gene, these are the 5 TFs, and then you give the input expression of those 5 TFs, and then you try to predict the gene expression. And that's your reconstruction. And then iteratively, the model just refines this networks, which best explains this reconstruction profile. Obviously, with the TF2 gene, it's not just any unconstrained optimization. There's a lot of constraint which are all inbuilt in the model, that the TF has to bind to a region an attack peak, which is an enhancer of the gene. It needs to have a motif and so on. So it's sort of like a constrained optimization. So you learn a GRN, predict expression, use that loss to learn a new GRN, and then do this iteratively until you converge. Yeah, if that makes sense.
And another thing is that we know that the GRN networks are huge. Like in your graphs, like you pick some specific nodes.
No, so basically, like every GRN is like a full network. So like its topics is TF to gene. So basically, you will have if a TF is not regulating a gene, that value will be 0. So you just think of it as a matrix, basically, yeah.
Okay.
And we have like L1 penalty in this optimization, so there's some sparsity and there's a lot of details.
Right. A lot of stars in the network. Yeah.
Yeah.
Thank you.
Yeah, no worries.
I'm muted. Great. Any more questions from the audience? If we do not, that ends our recording right on time, it seems like. Thank you, everybody, for coming out to the 32nd edition of the Multiomics Reading Group. And thank you, Manu, once again for the wonderful presentation.
Thank you so much.
Have a great rest of the day. See you guys later.